All right. Hello, everyone, and thank you so much for joining us today for today's webinar. My name is Courtney Wollstonecraft. I'm the Product Marketing Manager for iClicker. We are going to get started momentarily, but before we do, I just have a couple of quick announcements. First, we will automatically mute everyone to help keep out background noise. However, we definitely want to hear from you, so please feel free to type any questions or comments in either the chat or question area. I'll be monitoring that throughout the presentation, and then we can do questions after. If you'd like me to unmute you to speak, I can do that as well. So if you just send me a request in the chat area. We are recording this presentation and we'll share a link out shortly after it's finished. We also archive our webinars on iClicker.com so you can see a full listing of future webinars that we're hosting, as well as all of our recorded webinars. So with those things covered, I'm pleased to welcome our guest speaker, Deb Peers. Deb is an instructor for introductory biology curriculum at UCLA. She teaches courses in evolution, ecology, and biodiversity, cells, tissues, and organs, and molecular biology. Dr. Peers is a longtime Bruin. She earned her BSA in biology and later her PhD in ecology and evolutionary biology at UCLA. And Deb also, in her wealth of spare time, teaches at Moorpark Community College in Ventura County as well. So without further ado, Deb, I will let you get started.
Hi, thanks so much for the intro, Courtney, and thank you to all of you for coming today. I know that Fridays are kind of the day where I feel like I need to catch up on everything I didn't get done during the week, but I also have to get ready for everything that's coming up in the coming week. And if you're on a quarter system or a semester system, it doesn't matter. Wherever you are, there's always plenty to do. So I'm really thankful that you guys took some time out. I'm hoping that some of you are actually being able to enjoy this while, eating lunch, so multitasking, so to speak. And really, I'd just like to give a quick overview about what I'm hoping to accomplish. I think that we hear a lot sometimes from administrators, or we go to an education meeting or workshop, and everybody, active learning is kind of this buzzword, and it means different things to different people. And so what I just want to do is kind of give or present some ideas for how to kind of be more active in your instruction while you're with your students. So not kind of like the talk where you would ask a clicker question or two, because then you're using clickers and you must be active, but ways to be more active and to really have more intent with what you do with your students in your classroom to achieve a goal. And so the examples today are all biology, but I'm going to try to frame them in such a way that you can see what the purpose is of each one of the kind of types of exercises that I'm going to show you. Three of them, or the first three that I'm going to show you, are a little bit maybe more time intensive. And then I've got just a couple quick things at the end as well. So if you're not quite sure how active you want to be, and I don't know how active you already are, some little quick things that you can try just to see how it works out and find out what your comfort level is, so that then you can kind of build on that. So this picture that I have on the very front, or the very first slide, is this. I just love it because a big transformation happened for me in my career, and also in kind of my happiness with my career, when I decided that I wanted to go on a journey down a learning path with my students, and I wanted to bring them with me. And I didn't want to just walk along the path and point things out along the way and hope that all the students were keeping up and listening and paying attention and whatnot, but that really this path was something that I engaged in with my students, and it was something of an experience that we could share together. And when I kind of changed my mindset to that, it really helped me as an instructor. I'm not saying that that's something that works for every instructor. I just, want to make sure that people are kind of understanding why I do the things that I do in my classroom. For a lot of years, I've been watching report after report come out of, different government offices, keeping up with the literature and things like cell biology education or, excuse me, Journal of Higher Ed, going to different conferences. And really for, a little over 20 years, there's been a large discussion about active learning and that we need to engage our students more with the material that they're learning. And it's not enough to kind of construct courses where we might say, if I tell it to them, then they will learn it because it's important if I have told them. I guess we end up in a little bit of something where if we really believe that just because we say it, they will learn it, then we should just be in front of classes reading lecture notes or reading textbooks. And then because we have said it, they will learn it. And that isn't necessarily the case. And if we really want students to engage with material and see relevance in material, sometimes it isn't enough for us to say, this is really important because, right? Oftentimes what really makes an impact on students is when they're able to construct that opinion on their own. And we as instructors are also going to kind of have to let them go that they may see things our way or they may not. There are lots of student-centered teaching practices. This is out of something by Bill Wood from 2009, but this is kind of like a table that's a comparison in between traditional practices and research-based practice. And the way that the course is organized, how students get feedback, all of these things have to be changed a little bit in order to move from kind of a traditional practice to a scientific teaching, evidence-based research kind of aspect for teaching your courses. And there were a few things that always seem to be important across a lot of the literature that I've read, is that, it's not just about, a syllabus shouldn't just be about topics, but it should be about learning objectives. What do you expect students to be able to do when they're done with your course? And that assessments shouldn't just be kind of high stakes exams, just a midterm and a final instead, or a final paper and, another 2 exams maybe that make up their entire grade. That they need feedback, and students need some lower risk chances to kind of get practice at whatever skill it is that you would like them to have by the time they're done with their course. And one of the other things that actually kind of resonates with students and the success of active and student-centered teaching practices is that it's important that instructors can be accessible. And that becomes a little bit easier now with things like Zoom conferencing or setting up a Google Hangout. And the accessibility can change in that you don't always need to be in your office at a specific time. So there was a paper that was, excuse me, published in 2014, and it was a meta-analysis that kind of changed our conversation about how we should change our teaching approach. And this was a meta-analysis of over 200 different studies that looked at active learning and discussed how important it is that traditional lecture courses really don't do well, especially for our students that are traditionally classified as low socioeconomic status or as underrepresented minorities. Also included in those categories, things like first-gen students as well. And so those categories of students were actually identified by this study to be more likely to fail in a lecture course than they were in courses with active learning. Now, in active learning courses, we also expect that there's a little bit more structure to the course as well. So you would expect that there would be some pre-class assignments, and not only pre-class assignments, but that in class students are working with the instructor as a mentor or as a coach, as a study buddy, however you want to think about that, to kind of practice and master and also teach to other people, their students, their fellow classmates, how to achieve everything that you've listed in your student learning outcomes for your course. And I talked just a little bit right now about a really highly structured course, but the things in the course also need to be aligned, especially if you're going to have activities. When you're going to work with students, you're going to ask them to answer questions or talk to the person next to them. You might hand out half sheets of paper that are blank and ask them to write something down and actually turn it in to be evaluated as one of kind of a low stake assignment. And when I think about structuring a course in this way, I always think about alignment, right? What do I want my students to be able to do when they're done with the course? And I think about what are these things my intro bio students, left my class, and a year later, what's the thing that I would want them most to be able to do? Do I really want them to have memorized the difference in between two classes of sponges in diversity? And I think about it that way. No, I don't. That's not as important to me. Do I want them to understand maybe the ecological role of sponges in marine communities? Probably. And so I can really think about my course and reshape my course based on what are these fundamental broad concepts that are things that I want students to be able to do. And then I need to think about what will students have to show me? What evidence will I need to know that the students have actually achieved those outcomes? And in order to have students be able to show me, I want them to be able to give me that evidence, because that's going to be my assessment of them, and a large part of what their grade is based on. How can I help them? Kind of what can I design to help those students achieve those outcomes? And so what I end up with is this iterative process where I go through, what do I want my students to be able to do? And that's always where I start. Before I teach any course, I always start with the outcomes. And the outcomes serve the students as their guide. They know exactly what is expected of them from day one. These are on the syllabus. They go in front of, in the LMS, at the front of every week, your learning outcomes for the week. They go on any slide presentation or any handout that happens when we meet in class. But this is where I start, and it gives me my guide. Here's the road that I want to help my students walk down. Here's the path we're taking together. We're taking this path to this outcome. And I need to think about the assessments. What am I going to expect them to show me? And if I expect them to be able to show me these specific things, then they should have practice at that. And if I'm going to give them practice at that, what types of activities am I going to work on with them so that we can achieve that? So I always like to give a nod to cognitive science. Sometimes we forget about this in the biology education literature realm, but there's a lot of work in cognitive science that talks about the importance of repetition and interleaving. And I know that for some people that see these titles like this, well, repetition and interleaving, that's flashcards, that's memorizing, that's like, well, no, it actually can be something that's quite different. And for interleaving, I think that we actually all do this. We start with something small, and then we move on to maybe another topic, and then we go back to that original topic, or we build on that original topic. And so interleaving is when you bring a topic in over and over and over, over some prescribed time. Retrieval practice, i.e. testing, is really important. Having pre-class quizzes, having post-class quizzes, having little quick, do you remember this, or try to draw this figure, or give me a representation of something. The more that students do these, the more likely they are to master concepts and to be able to integrate different topics. And I do have a lot of assignments like this, but they do not amount to a whole lot of points. Any individual one assignment is not worth a lot of points. But over the course of the quarter, a lot of these little low stakes quizzes and things like that are probably about 7 to 8% of their grade in the class, along with other things like practice exam questions and going to discussion sections. Those are all in between 8 and 10% of their grade as well. And then we have to, provide enough time for students to be able to ask questions about things. And if they're going to ask questions, we need to also answer them. So when we're going to go about activating our class materials, a lot of people think that they need to start from scratch, and you really don't need to. There's actually a couple different paths that you could take. Or you might get to some type of in-class activity. And the activity part is really important, because this is how you can kind of maintain being student-centered, and how you can kind of walk down that learning path with your students. So instead of being the sage on the stage, you have the opportunity to be the guide on the side. And really, again, goes back to the learning objectives that you can see up here. Is the material necessary to support our course learning objectives? And I whittle everything down that we do in class together is based on those course learning objectives. If there's background that they need and the students can do it on their own, then I make it an out-of-class activity, like a pre-class reading assignment or something like that. And then I work on what are we going to do in class together. I try to have activities that are kind of grounded in Bloom's taxonomy. And oftentimes in these activities, I start small. Small is probably the wrong word to use, but I start with a lower order cognitive skill. Whenever I start with an activity, and I begin with questions, either clicker questions or with whatever they're doing on a handout. And I ask them more of kind of, do you remember or understand this concept from your pre-class assignments? And then we kind of work through the higher order cognitive skills when we're in class as well. So early questions in an activity, which I'll show you some examples in just a minute. Early questions in an activity might be a little bit of a lower order cognitive skill. Have them kind of reflect back on what they did before class. So I know that they have the foundational information necessary to be successful in working through the activity. And then in the activity, we try to develop higher order cognitive skills. And if you're not familiar with this presentation of Bloom's taxonomy, or with Bloom's taxonomy at all, you can just Google it and you'll find more than enough in terms of images and sample verbs that you can use to be able to kind of define your student learning objectives, all based in Bloom's taxonomy. And that's something that would help you perhaps design an activity for in class. So there are some simple things that you can do, and I'm going to give you 3 examples. Again, they're biology examples that we do in courses on this courses in our introductory biology series. And I would like you to know that I use them in the classes that are taught at UCLA, as well as the ones that are taught at the community college. We are equal opportunity learners, and I don't think there's any difference in between students. And so this is probably one of the things that has really helped me as well, is to be able to think about, you have to, one of my personal goals is to bring everybody up to a certain level. And so while that means that you might start lower than some students' levels, you're starting right in the right spot for others. But we all do better when we all do better. And so bringing everybody kind of up really helps the class kind of participate, and they have a shared experience, and there's no competition. It's about everybody kind of making it through the activities. And so one of the things that I like to do, I have a lot of half sheet handouts that I make. You don't have to do that. The other thing that you can do is whatever data you're going to put up, you can leave that on a document camera or just on a slide. And just leave that up during the activity. I like to do half sheets. Sometimes they're blank on the other side. Sometimes I have something else, some other activity that's there on the other side. But I like for students to be able to kind of take a look at some data, and then I have them discuss with their neighbor. And this gets loud. So my classes are, well, one of my classes at the community college are 24 students, and my classes at UCLA are 435. And it is an absolute cacophony when this first goes up. But after about the first day of teaching, my students know that I really want them to talk. And I invite it, and we have rules. So there are signals for when it's time to stop talking, because you can't wait 10 minutes for 430 students to stop talking. But this opportunity for them to speak in class and discuss is one of the things that comes up in the highest frequency on evaluations, and not in a bad way, but in a good way. Where a lot of students acknowledge that they feel like they learn better when they're able to say things out loud and explain things. And they liked having the opportunity to do that in class. So this is a kind of a gene expression, looking at histone or DNA modifications. And this first discussion thing is just you give them data and you let students talk about it. Have them jot down the answer to these questions. And I don't, this is not something that I give them 15 minutes to do. I give them, anywhere between 3 and 6 minutes, depending. I walk around and see what they're doing. And after, I'm looking around and I see that this is done, I don't then explain these figures. Instead, I go straight to a question. And if you use clickers, then this can be a clicker question. If you don't, then you can ask students to raise their hands. You can do throat votes. You can do color cards, whatever. But I don't say anything in between when I put up the signal to kind of quiet students down, and then I move on to this. Their discussions are what I am hoping will let them answer this question. If you do use clickers, then you can look and see how students are doing with the answer to the question. And if they're really far out, or you end up with 30% or 33% for choice A, B, C, well, then you can take a step back. You can see that your students are struggling, and you can, in real time, re-engage them on the learning path about this concept. Then there's other things that we can do. So this question is a kind of a lower order cognitive skill, right? So it probably borders in between lower order and higher order with a little bit of interpretation. But now we're going to ask them to incorporate, integrate, and interpret another bit of information, and then ask them to evaluate a new set of data, which is what shows up here in this gel, which we're calling a northern blot. I know that many people are advocates for only real data, but sometimes you can't always do that. And this makes it a little bit easier, and you can move to real data at another time. And then we can give them the answer, and depending on how they were going to answer, you can see how they're doing it, being able to integrate and things like that. One of the other ways to kind of activate your lectures is to provide a scenario. And I do this an awful lot in the physiology sections of my courses, but also in the gene expression sections of my courses. And there's just endless possibilities. And I mean endless possibilities for manipulation, which are just, it's fun, This was, we just had the Olympics. We're talking about, even though this is not a Winter Olympic sport, obviously, but talking about athletes and athleticism and what can they do. And so the instructional team and I decided that we might want to do something like, let's talk about marathoners and what actually happens during a marathon. And so again, they have a blank sheet of paper, and the first thing we ask them to do is describe what you think a marathon runner experiences over the course. They've had pre-class assignments, they've done reading, they've had some kind of recall quizzes, basic understanding questions. And so now I'm asking them to say, what do you remember from that? So if they finished it on Sunday, maybe in classes on Tuesday, they don't remember that much about it. Or if they just finished it right before class, they might remember a lot. But again, that active recall goes back to what cognitive science tells us is something that's really important for students having long-term conceptual understanding of a concept. So they're doing this, and I say, well, how many of you said that they're probably going to drink something? And this is just a simple hand raise, or you could do it as a clicker question. Again, that part doesn't matter, but I get some idea of how many people thought about, some people go to, the marathoner in third is really just worried about being in second and then in first. And other people talk strictly about the physiology of muscle shortening and the mechanics of that. And some people remember to talk about maybe they might take in some water. And so we, in this time, I can say a little bit about water and the importance of fluids, and I can ask students to say something about where they think fluids can be gained or lost during a race. Just a general conversation. And what this allows me to do is to kind of increase the comfort level with asking questions in class, but also saying that I'm just as willing to engage in conversation with my students as my students are willing to engage in conversation with me. I usually give a quick little review, especially for something like the nephron. And if you don't teach a physiology section of a biology class, that's fine. Then you're probably thinking the same things a lot of my students do, which is, oh my gosh, there's so much, right? And this is one of the things that happens with some figures. It's just there's so much. It just looks like I'll never be able to get it. But there's a way for us to kind of think about conceptually what is happening. And so I think about how can I ask questions to help them conceptually understand what is happening in this system, what's happening in the nephron. And so these are just, I might give just a general little like 3-minute overview because I've made my students accountable. They've already gone over these figures. I've already asked them some questions about the nephron. For some of my classes, I have what are called reading guides. And so they might have brought their reading guide to class so that they can just concentrate on some, but not everything that's inside a chapter. I can actually help them direct their pre-class efforts with a reading guide. So they might have that to refer to as well. And then just, the questions begin. And if they can remember, point F, that this is the collecting duct, then I like them to be able to use these terms appropriately. And this is really a remembering or understanding. It's a lower order cognitive skill question. And I always have the answer be the second slide in case I have to re-pull while I'm using clickers. But then I can ask something a little bit more involved, and this would be a higher order cognitive skills question. And so I think that one of the things that's always important to remember is that it's good to kind of move through the levels of difficulty of Bloom's taxonomy with your students in class. So it's very seldom that I would just ask this one question about the nephron, and then I would move on to something else. And the samples of questions that I'm showing you here actually are not all of them that I would do in a single class period, but I do go through from kind of lower order, more understand, remember questions to let's apply and let's integrate, because on exams and my student learning objectives are primarily higher order cognitive skills. And so I can look at the cells around region B, and then for follow-up, I can ask a question. So would they mark A for true or B for false? If maybe not as many students as I wanted to did as well on this question, or maybe not a lot of them got it, then I would kind of take a step back. We could still have a discussion. I might draw something up on the board and help bring up a few more students to the place where we would like them to be on the topic. But then I can also follow up and say, what region, which letter do you think you would expect to see more sodium potassium pumps? and I can have students answer that, and then I can show an alternative picture. And we can talk about, we've talked about membrane potentials in the sodium-potassium pump, but they're not just in nerve cells. And sometimes students tend to forget that, or this is a topic that's in the third quarter, and they actually first heard about sodium-potassium pumps in the first quarter, and so it's a good chance to also be able to remind them. We can talk about cell polarity. Lots of things can happen. And then I can move through this even further and talk about where do the things that were reabsorbed go. There's stuff that's missing here. And in this figure, things that are important to a student in physiology aren't showing, like the circulatory system. And so I can use questions to ask them to recognize what is missing and what is apparent. So what is this figure actually showing me, but what is also missing? And we can talk about why nephrons would be so heavily vascularized. We can, and what are they picking up? We can move on to kind of a whole body experience and how are we gaining water, how do we lose water, and turn it into something that's a little bit more comprehensive. So although we started very small in scale, you can move up in scale. And again, this is all aligned with the learning objectives. And then I ask about, here's the bigger one, and this is what I want you to think about before you get to lab. Sometimes I have people think about this, and it's the first question we'll come to at the next class meeting, so we can come up to that again. And this is where you actually have them making predictions. So again, that's a higher order cognitive skill. It will be one of their learning objectives, and they can do that. Jigsaw activities, I'll be honest with you, I love them, but they can be daunting. So I would say that jigsaws are not for the faint of heart, but I do think, especially if you have an upper level class, or if you have a class where you like to look at a lot of data from primary literature, then jigsaws are really, really great. And this is based on collaborative learning, and it emphasizes group work during a class meeting. And so the way that jigsaws work is that you want to kind of define a problem. And the question that we're going to try to answer with this jigsaw is, what is a consensus sequence? We can look at different consensus sequences. If this is not material that you teach, it's perfectly fine. I don't expect everybody to know these types of examples. But you can say that there are kind of differences in what happens in whatever question it is that you're trying to answer, if there's a mutation or something like that. And then you can set up a jigsaw that will try to answer the original question that the class was addressed with when they first came in. And so this is one actually about how well translation efficiency was, how well translation actually occurs, transcription, and translation will actually occur in individual organisms based on a sequence. And in this case, they're looking for the start codon, which is related to translation. And in jigsaws, you can have 1, 2, 3, or 4 phases. Most of them have 2 phases. And you need 2 different worksheets. And in this case, a worksheet would be a piece of data. And so one group of students, and this is usually by row, gets 1 piece of data, so 1 figure that they're going to have to interpret and explain back to each other. The next row gets the other piece of data, and they're going to work together with other people that have the same data as themselves. Once they've worked on their worksheet and they've answered the questions that are there with their classmate partner, and you're also walking around to help, then they're going to turn around. And so in phase 1, they talk to somebody that is looking at the same figures and data that they are, but in phase 2, they're going to turn around and talk to somebody else. And in this way, they have to explain the data that they looked at with their first partner, and now in this new group, they have to integrate the information that was given to them. And so Oftentimes I like to show the handout that's up on the slide. If you see that I have an A in a circle up in the left-hand corner, that means group A, if you have this, this is what you are doing. And these are the questions that you will be answering. And then there's also B, and I say B, these are your data, and these are the questions that you are answering. So do that with your partner now, and in 5 minutes you're going to switch. And then after both groups have met, then I can again ask a series of questions and have them put it all together and have them write down the answers to this. And then typically I go straight to the clicker questions. So I use the results from the clicker questions to gauge just how much discussion or follow-up questions I might need to have with my students. And then I give them, you know, here's what we actually do know, and you just figured out what most textbooks take, you know, 5 pages and 3 figures to teach you. So good job. I like to tell my students good job a lot. There are lots of different places. There's actually a Jigsaw Newsletter. So if that went a little bit quickly, or you're interested in trying this, these are jigsaws, or this website is all about jigsaws for ecology education. And it is just a great site. It's has a lot of ideas, you know, and some of them I use just as is, but other ones that I've used, I've edited a little bit, and it's always easier to edit than it is to create. And not all data, you know, or research experiments are really designed to be part of a jigsaw. Okay. And I know that some of those probably seemed a little bit involved, very involved maybe. And you just want to be active a little bit. I just want to try it. I just want to see what happens. A lot of faculty that I work with are maybe uncomfortable with the free form of having a lot of student discussion or a lot of clicker questions or having handouts in a large class. You know, believe me, there are days when I'm not even sure that it is remotely organized chaos in my classroom, but my perception of it is usually far worse than the students' perception of it, where they really are just enjoying this learning process. But there are some other ways to just, you know, dip your toe in the water a little bit, try it out. And so I'm kind of moving away from the more involved to just some quick examples here about how you can just ask a question or two in each class meeting that is a little bit thought-provoking, is relevant to the topic for the day. And it might be something that students haven't thought about, but makes them think about things. So maybe before your lecture about fungi, you could show them this picture and ask them this question. You know, how many individuals are actually present in this picture? And you can use this and the answers you get to kind of guide or have something that you can refer back to over and over and over again during your class meeting as you're talking about, you know, the anatomy of a fungus. You know, what's a hypha? What is a single hypho? What is that mycelium? How does it grow? Why are these primarily in the shape of a circle? I mean, lots of things like that. And as you're going through a kind of a fungus lecture, where you're trying to get some information to them, you can keep referring back to this picture, and you might even paste this picture into multiple slides throughout the day, and so that you can, after you've kind of explained something or described something about fungus, you can come back and say, and actually this is evident in this picture, you can see it here. And so it's a way of kind of starting students off. You get them thinking, you get them going. You realize that you don't learn everything just from reading a textbook, you know, and you've got them curious. You know, a lot of students might look at this and say, oh, I've never really thought about why fungi grow that way. You know, so after you have those follow-up, after you have that initial question, there's lots of room for follow-ups. And letting the conversation morph is really a good way to begin to either frame what you might do the next time you teach the class, or maybe work in something else about fungi in a later section of the class. right? So you can come back to that topic again, and then that helps with the interleaving that I talked about earlier on today. Survey data is great, especially in just about any class. So this is not just for science people, but you could, look up surveys on, which, I'm trying to think about one, like how many people surveyed recognized that if you were on the moon and you were holding a pencil and you let the pencil go, what percentage of people said that it would fly off into space. You could look at those common misconceptions that are societal misconceptions about basic concepts in science, and you could ask your students the same question. And so what I like to do with survey data like these is you get your survey, and before you're going to show those survey results, ask your students the same question. And then over the weekend, compile them, kind of make the same graph. And then in the following class meeting, you show the national results. And then you show your students the class results, and you ask them to compare. And you simply ask them, why do you think these differences might exist? What are similarities and differences? Can you describe the data? And so again, students are engaging in things. They're looking at, the possible relevance of a topic. But, there's a lot of things that could be done rather quickly, and show students kind of how to use data to be self-reflective of their own answers. But also, anytime you can get data into a science class is a good thing. The best ideas come from your environment. Questions in office hours, that student that says, well, what if this were to happen? In office hours, you would say, well, what do you think would happen? Let's talk this out. Turn that into an activity. That idea, that is the nugget of something that can grow into a beautiful activity, and then everybody benefits from watching you model a pattern of logical thought, for problem solving. Written responses on homeworks or labs or discussion sections. Sometimes students write down their reasoning, and it makes misconceptions very easy to see. And so designing an activity that's explicitly for explaining, or that's explicitly for addressing that misconception can be important. I talk with other faculty. I'm lucky. We have 3 courses in our intro series, and we have stewardship committees for every single one. So the faculty that are going to be teaching in that class for the quarter meet every week for 2 hours, and we go over what things we are doing and what worked and what didn't work, because we have multiple sections, and most of these use all the same examples. And we can talk about how we need to tweak an activity, which ones we just need to throw out because they're just not accomplishing our learning objectives. And every once in a while I'll be looking for something on the internet and I come across something else, and I have a file on my desktop where I just throw figures in there or jot down ideas. And when I have time, I kind of work those up into more involved things. But I would say that, when I come back and look at this list, this quarter it has been questions in office hours. I've had a lot of students that have been giving me the what if scenarios. And it always makes me smile a little bit, because then we get a chance to say, well, let's solve that problem. Let's think about what would happen and what kind of information we would need. And so, it's nice that there's a website about jigsaws. It's nice that there is the National Center for Case Study Teaching. It's nice that there are activities that are included with instructor resources and some textbooks. But there are a lot of great ideas that we hear every day if we're receptive to hearing an idea about a potential in-class activity. So I'm ending with this slide so that everybody can kind of think about where they might start looking for a potential in-class activity. I have the chat window open and I don't see any chats. I'm not sure if any questions have come in, But I am more than happy to answer some questions now, give some ideas, or show you anything else if there's something else that you would like to see.
Yeah, thank you so much, Deb. This was really incredibly helpful. A lot of helpful tips that were specific to biology and some that were very applicable in other disciplines as well, which is awesome. I do have a couple of questions, and I want to encourage everyone to, if you have other questions, to type them in the questions box or the chat box, and we'll be happy to get to those. But let me just start with this one here. You talked about a couple of different types of activities. Which activity types do students typically respond most positively to, or have you seen, I guess, a more positive response to certain types of activities from you?
Yeah, so I would say that the ones that are most, I would say the ones that this students will tell you are most successful, I like the ones where they look at data and they get to talk about data with their neighbor first. And then without talking about it, when you go into asking questions and clicker questions, then the students are excited to see how well they did kind of on their own, understanding the data that was being shown to them. I will say that those, when you look at exams and exam performance, those are really, really highly correlated. Those questions are highly correlated with data interpretation questions on the exams too. And so I think that over the time course of a quarter, students really start, after the first midterm and they see how well they do on data interpretation questions, they're ready for more of that. But sometimes it's hard, I'll be the first one to admit, sometimes it's hard to find data. Sometimes you can't think of exactly how you might want to make a bar graph. This is where a question bank or how do we know figures in a textbook or things like that are really, really great to use in class.
Yeah, that's a great point. How do you create the outcomes for your course? And I know you talked about referencing Bloom. The second part of that question is, are they consistent across the department at the schools where you teach?
Yeah, so the learning outcomes for the course, it doesn't matter where I teach the course, right? So I try to keep that the same. Okay. So the objectives really do come down to, when I would just do straight lectures, I'm going to be honest with you, I would do a lot of stuff that was like defining terms and pointing out labels of things. But on the exams, I was killing my students because I was asking them to do all this stuff that I had never asked them to do before. And while writing those questions, I would say if they really understand, osmosis, whatever, insert topic here, then they should be able to answer this question. And so when I first started getting all the objectives together for my course, they've been refined and edited and changed. And I've added some and I've taken some away. I went back to my exams and I said, these are the things I was asking my students to be able to do. And I just turned them into a more formal statement, a more formal language, using verbs from Bloom's taxonomy. So that was where I started.
Gotcha. Okay. Okay, good. And then just one more, how that I'm seeing, but feel free to type some more in. How long did it take for you to implement changes in your class and to make it more active? And I know, I guess it's probably a gradual process, I imagine.
So, it is a little bit, so I would say that for a little while, I had an active classroom that was really instructor-centered and not very student-centered. And I would ask a few clicker questions and I would feel better and pat myself on the back for being active. And I think that for me, it was an absolutely great start. But I would say that it's been the past 5 years that it's taken for me to be, I now have what I think most people would consider a flipped format classroom, although I don't require watching videos. They don't watch lecture videos before class, but they do have to read and they do have to take pre-class questions. But now, I have probably 2 to 4 activities per class meeting, and anywhere from 15 to 20 clicker questions and 2 handouts. And my class meetings are over an hour, so they're not a 50-minute class.
Okay. Okay. Well, I think that's it. Thank you, everyone, and thank you again, Deb, for joining us today and for the wealth of really helpful information. Again, we will have this session recorded, and that will be sent to you in short order. And as a reminder, you can also access our full listing of webinars for the remainder of this spring on iClicker.com and inside the Learning Center, which is on the top menu bar, as well as all of the recorded webinars, which is where this one will be added as well early next week. So thank you all so much, and have a great rest of your Friday and a great weekend.
Thank you very much, everybody. I hope that you guys have a great weekend.
All right. Thanks, everyone. Take care.